All right. Let me just share my screen here. All right, so I assume everybody can see the screen okay and hear me fine? If
Yes.
not, just shout out. Okay, excellent. Yeah, so as Trupti noted, I I am the director of the Smart Core, which is really kind of a place that does some of the newer genomics kind of technology. Today, I'm going to focus on three different methods for what I call transcriptome-wide profiling. And so I'm a physiologist. That's how I was trained. That's my PhD. And then now I'm more of a cancer biologist. So that's kind of the perspective that you're going to get. I'm not a hard line kind of genomics gene person. So I come at this as a little bit different in terms of big picture, what these things can do. And then what I hope to do is kind of highlight what the core can do for you. And man, it seems like it's advancing. So, all right, we'll go for it. So I'm going to start off with bulk RNA sequencing first. It's the first kind of technique that came out that allowed kind of global transcriptome profiling. It's my favorite one of the ones that we're going to talk about today because it's highly useful. It's very affordable. It's something the core can do readily, and there's established pipelines for this. So you get your data, in a very quick fashion compared to some of the other techniques. So again, I'm not sure what everybody's level is throughout the network. So I would really wanted to kind of step back and talk about, when would you actually use bulk RNA sequencing? How would you incorporate it into, what you're doing? And really, what it boils down to is if you want to determine global gene expression changes, so all the different mRNAs that are changing in response to some experimental manipulation. So it could be a hormone treatment, a drug or a toxin, relative to a control group. So that's the type of information that you're going to get. And the reason I like this so much, and I'll provide an example of a paper that we had, a couple of years ago now. But when you take your RNA expression changes, so all the different mRNAs that are changing, and you apply pathway analysis to that, what you can actually identify is a readout of cellular pathway activity. And I think that's really kind of how I've used it in the past. And I think that's a strength of the assay. And again, so it'll identify differentially expressed genes in response to some experimental manipulation compared to a control. And then with the right pathway analysis, you can identify functions and pathways based on those expression changes, using an unbiased approach. And so this actually becomes important if you have a compound, a natural product, for example, that is having a cellular effect, like for instance, inhibiting cell growth, but you have no real prior knowledge about the mechanism by which it's doing. And one way to address that is to, identify the differentially expressed genes that are changed by that natural product, for example, and then taking those gene sets and then asking what pathways are they associated with, and that can get you a step in the right direction. And how we've used it in the past and how a lot of people use it is an excellent hypothesis generating tool. So I think the best way of doing this in terms of showing, how you can use this technology is to use an example that we actually published, almost 10 years ago now, in which we identified a gene was being regulated by the aryl hydrocarbon receptor. So, just from some interest perspective, in terms of, the kind of things that we used to be interested in, we were interested in a ligand-activated transcription factor known as the aryl hydrocarbon receptor. And because it's a ligand-activated transcription factor, it was perfect for an RNA-seq experiment because it would induce changes in mRNA. And just out of interest, so you can kind of see the flow of this, the first aryl hydrocarbon receptor ligand, again, which is a transcription factor that's activated by a ligand, was actually an environmental pollutant known as TCDD. And so, to really kind of appreciate this, TCDD was actually a component of Agent Orange, which was used, years ago to defoliate forests during the Vietnam War. And what occurred is that as they made Agent Orange, this herbicide, one of the byproducts was TCDD, also known as dioxin. And the reason this is important is that, post-war, some of these veterans and other folks that have been exposed to this had increased risk for certain cancers. And, at that particular time, we didn't understand, the compound that was increasing the risk for cancer in these veterans. Now, it turned out to be one of them was TCDD. And so the question is, well, how do you generate it? So this is the agent, the herbicide. And what occurs is if you heat this up too much during the processing of it, you will then generate this compound, TCDD, or 2,3,7,8 tetrachlorodibenzoldioxin. And the reason that this is was such an important chemical at the time is that it stays in the environment for up to 100 years. So it's still there, and it accumulates in soil. It gets transferred between countries in the air. Whoop. For some reason, this is advancing. I don't know if it's catching my movements, but nonetheless, let me just finish this up. So it stays in the soil. It gets in humans. It stays there for almost a decade, and it's resistant to breakdown. So once you have it, you have it. Now, the reason that we became interested in this, and again, I'll show you how we found some interesting things using RNA sequencing, is that the types of aryl hydrocarbon receptor ligands that are present are not just TCDD. So it's present, these compounds that can activate the AHR are also present in cruciferous vegetables like broccoli, indole-3-carbinol, and these are in fact good environmental AHR ligands. And it's also produced endogenously. So there's kynurenine, which is an endogenous aryl hydrocarbon receptor ligand. And then there's certain drugs that are also AHR ligands. So there's a lot of interest about the AHR. And so that led us to our question. So we wanted to investigate, how activation of the aryl hydrocarbon receptor would change or modulate the cancer process. And again, as I noted on the previous slide, there are lots of different HR ligands, but what we were interested in is TCDD because it is the prototypical HR ligand. So its effects are almost exclusively mediated through the aryl hydrocarbon receptor, making it a really good experimental tool. And so when you think about what it's doing in terms of framework for our hypothesis, and if you're interested in RNA sequencing, you would think kind of along these lines, is that it changes cellular physiology or cell biology by regulating gene expression. So it travels into the nucleus, it'll bind to its regulatory elements, it'll induce changes in mRNA, which will then induce protein changes. And again, at this time, when we did this study, we knew that dioxin increased cancer risk, and we understood, that this is important for veterans and other folks, but we didn't understand the mechanism. So this was a kind of a first approach at, getting at the mechanism of action. And so again, the hypothesis is that TCD would go into the nucleus, regulate changes in multiple mRNAs, which we didn't know at the time, and that these changes in mRNA would then reflect changes in gene activity, and then we could then map back in terms of the mechanism of action of TCD based on the mRNA changes that it was inducing. And when you think about bulk RNA-seq in terms of what the core will do for you, it's kind of highlighted here. So as an investigator out there in the network, you would extract RNA from your experimental group. Right? Ours was straightforward. It was either vehicle-treated cells or TCD-treated cells. Once we kind of verify that the RNA is good quality, we do QC analysis here in the core. Brittany would then build sequencing compatible barcoded libraries. And so that's a kind of a sophisticated technique in terms of successfully doing this. And Brittany's surgeon, who's the technician in the SMART core, is very good at doing that. So then what happens is you take these barcoded libraries. So they're barcoded, meaning that you can then identify which samples those RNAs are coming from. And that's critical, because if you don't know that, then you're not going to be able to make any sense out of this. So based on those barcoded libraries, you can then demultiplex it. So you can then separate out where these RNAs are going. Are they in the vehicle groups? Are they in the TC-to-D groups? Okay. And then you have somebody do some bioinformatics for you. And what you're asking for is the piling up of sequence, let me just go back, on a particular gene compared to a control gene. So this would be, gene A would be a gene that's induced by TC-to-D. All right. All right. So a simple approach. We had 4 plates of control, which is just vehicle, and then 4 TC-to-D, and we just treated for 6 hours, which is not a bad time point. So as you think about your own experiments, if you go too long, so if you have and I'll just use natural product because I know some folks are doing that, and you treat for 48 hours, for example, you're going to have a lot of, initial changes and then changes based on those changes. And so you're just going to have a lot of changes that you're going to have to kind of sort through. So if you're really interested in kind of an acute response, which is what we were interested in, you would treat for an earlier time course, like 6 hours. Okay. All right. So we did the experiment, and what you would get are genes that are increased and mRNAs that are decreased. And that's exactly what we found. So again, this is a very selective chemical. So it only works through the aryl hydrocarbon receptor. So these genes, we interpret this as being dependent on that ligand activated transcription factor, the aryl hydrocarbon receptor. Now, when you get that kind of data, so again, it could be a natural product, or it could be a specific ligand for a transcription factor. The next question you're going to ask yourself is whether or not the mRNAs that are changing, the genes that are changing, are they in fact direct, in this case, TC-to-D AHR gene targets, meaning that the AHR is actually binding to a regulatory element in those genes to induce changes in mRNA, or what you're actually observing is an indirect effect, in that the compound is inducing some effect somewhere else, which then is having an indirect effect on those changes in mRNA. So you will face the same kind of question. Now, you can get at that question depending on what you're investigating. Now, we were interested in a ligand-activated transcription factor, the aryl hydrocarbon receptor. So what that means to us is that we can actually identify direct AHR target genes by using an approach known as chromatin immunoprecipitation. And what this does is it freezes proteins to DNA because you fix them in formaldehyde. And what you then do is sonicate that, and you're interested in the sequence that the aryl hydrocarbon receptor is bound to, right? And then you immunoprecipitate out with the aryl hydrocarbon receptor antibody, beads, and then what you do is you release these very specific pieces of DNA that come out with it, and then you build sequencing compatible libraries. And you ask what genes are actually bound to the aryl hydrocarbon receptor. Now, from a core perspective, ChIP-seq is a very difficult experiment. You as a laboratory, as a researcher out there in the network, you would be responsible for the actual immunoprecipitation. And then as a core, we would verify that your experiment has worked. And you would definitely have to talk to Jim Dinbier, to make sure that you have all your experiment in terms of controls and things set up. So it's difficult to do, but it's something that we've done in the past. So here's the exciting part. So this is why this paper was exciting, is because we started off with 663 genes, and we wanted to identify which of those were direct targets. And so what we did is we overlapped those mRNA changes with genes that the aryl hydrocarbon receptor bound to. And by doing so, we actually got a very specific set of genes, 108 genes that we reasoned to be direct aryl hydrocarbon receptor gene targets. So, allowed us to get a mechanism in terms of how this signal is flowing through the cell. So here's these genes that we identified. A nice thing about this data set, and you would do the same on your end, is, do you have positive controls that are actually, in our case, truly TCD regulated genes? And we did. So these are the CYP genes, and you can see they're highly induced in the cells that got dosed with TCD compared to vehicle. All right. So this, in all purposes, this seemed like an experiment that had worked. All right. So then, the question becomes, with bulk RNA sequencing, that you're going to have, all these mRNAs that are changing. And if you're fortunate, you will then identify direct targets if you're dealing with a transcription factor or some type of histone modification. So is there a question out there? Okay, I'm gonna keep going. I heard, I don't think that, all right, so nonetheless, so then you ask it, what can you do with these genes? And that's where this pathway analysis will come into play. So if you do bulk RNA sequencing, again, the workflow is you generate RNA, Brittany makes libraries and generates differentially the mRNAs that are changing. Then you do bioinformatics to identify those genes. And then the next step in your progression is to do a pathway analysis on those mRNAs so that you can identify what pathways they're associated with. Now, at the time that we did this, we used Ingenuity Pathway Analysis, or IPA, which actually I loved. I thought it was an excellent software system to do these types of analyses. And I think there's, we're using different ones now. And again, there's folks in the network that can help you work through your expression changes, all right? But in our case, and then what you would do on your end also is ask what genes or what pathways are these genes associated with. All right, so then you get tables like this, and you can ask different questions. So here we were interested in what diseases were associated with the 108 genes that were changing. All right, simple question. And so what it's showing you are the number of molecules or genes in that data set that fall into a category, a disease category. All right, so 104 of the 108 genes were associated with cancer, and that's not too surprising because cancer is a big disease that has lots of genes and lots of papers that have linked genes to the cancer process. So this is all based on curated data. So this is pulling from publications and those kind of things. Okay. So we got some interesting other associated processes associated with these genes, but I think the power of it is this. So down here at the bottom was tumor morphology. So this was not a process that was linked necessarily to the aryl hydrocarbon receptor. So it identified it as being a process associated with those genes. And you can do even better because you know exactly what genes are being associated with tumor morphology. So I think that's the power of bulk RNA sequencing when you pair it with pathway analysis, is that you can get a very complex series of genes that are changing, but you can work your way down to kind of a meaningful next question. You can do other things. So that was a disease pathway. You can also ask for crosstalk with your pathway of interest, ours is TCD, with other pathways that are working throughout the cell. And in this case, this was an upstream regulatory analysis, which would identify genes that are being induced by TCD is also being known to be induced by another pathway. And then now they're being co-induced by both of these pathways. So it's a way to think about crosstalk. So we did this analysis, and here we looked at all 660. And again, what I would argue is we got remarkable data, and you would as well. So let's say you have a natural product and you don't know what pathways it's influencing or regulating to carry out the biological effect that you're seeing. Well, you could do the same type of thing that we did, and this is what we found. Nice thing about this is you may or may not have built-in controls. So what the analysis is identifying is 143 genes within the 660 that are being changed is already being shown to be associated with the TCD pathway. And that's statistically significant. So that is a nice positive control. It's also known that TCD interacts with the the estrogen pathway, and that's being highlighted here. But where we took this data is, and we published subsequent papers on this and wrote grants on this, so this is why this is so a powerful technique, is there was been a lot of the TCD regulated genes were associated with the Notch pathway, which is a developmental pathway that gets abnormally activated in cancer. This was the first link between HR signaling and Notch signaling, and it then led to additional studies, additional grants, and additional papers. So again, you can do this with your own type of experiments. All right, so now I'm going to move into single cell RNA sequencing. Now, this will ask a different question. So it's not necessarily whether or not one type of technique is better than the other technique. It's definitely not that. It's whether or not one technique will answer a specific question for you that applies to what you're studying, okay? So if you're comparing vehicle versus multiple treatments, I would argue that RNA sequencing would probably be the way to start. But if you're interested in something along this line, let's say that you have a tissue that you're interested in. It could be a piece of tumor tissue, or it could be a developmental tissue. And you know that that tissue contains multiple different cell types within that tissue. And you hypothesize that there are more cell types in there than what has been published. Okay, so you're looking for potentially another cell type in your tissue of interest. Well, there's a couple ways you can do that, and one of those is to use single cell RNA sequencing, and I'm going to show you why in the next couple of slides. But this is advertised, and this is what it can do. So it can reveal cellular tissue heterogeneity. So we know tissue is heterogeneous, has multiple cell types, and you're hypothesizing that among the ones that have already been reported, there's some new ones in there, and you want to identify what they are. And, so you're looking for potentially a rare cell population. And this approach can get at that question. So let's show you how. Now, half of my screen is covered up by the Zoom. So that's kind of why I'm losing some of these things over here. But nonetheless, hopefully you can see the whole thing. All right, so when do you need single cell resolution in terms of RNA sequencing? All right, so this is the question. So let's say you have a complicated tissue and you are proposing that there is an enrichment of a particular cell type that may not have been reported, or you are just interested in the complexity of that tissue to begin with. Well, what you would do then is you would dissociate this into single cells, which I'll show you here in a few more minutes. And then based on those, you're going to isolate transcripts that are unique to each of these given cell types. And the reason that's important in this context, so let's say you're just simply interested in knowing what cell types are in this tissue. If you did a bulk RNA-seq experiment, which wouldn't be applicable to this, if you just took that tissue and you just did bulk RNA sequencing, you just looked at mRNAs, you're not going to see any real changes because one, they're all being averaged, and then two, you're not comparing it against another experimental group. So this is why bulk RNA sequencing would not work for you, right? You want to know what cell types are in that particular tissue. And again, so what you would see is something like this, or one way to kind of visualize this is this. So you would have gene A, for example, or gene B. And those two genes are just expressed in this kind of purplish looking cell, and they're not necessarily expressed in these other ones. So this single cell sequencing would be a mechanism to enrich that and to identify that differentially expressed gene relative to the other cells within that population. Right? So that's kind of the goal of this, and I'll show you how it works here in a second. So really, if you're interested in identifying a rare, unique cell population based on shared gene expression profiles, because that's what this is doing, this expression profile is unique compared to these other cell types, then single cell RNA sequencing might be a technique that you would want to use. All right, so I'm going to minimize this. Now, how does it work? All right, so what would you do on your end as an investigator out there in the network? Well, what you would do is you would collect your sample and you would process it. Okay, so the genomics core wouldn't, the SMART core wouldn't be doing that. once we identify that, this prep is good enough to move forward, and again, we're going to show you how that looks in a minute in terms of pitfalls that you need to be watching for at this particular step, you would then give the extracted RNA to us, and no, I'm sorry, yeah, that's what you would do, and then we would prepare your single cell library using the protocols in place. You would sequence it, and then you would get an analysis, which would be kind of a lot of bioinformatic analyses. Okay, so that's kind of the overall scheme. Prepare the sample, make the libraries, sequence it, give the transcripts to your bioinformaticist, who then will do analysis and make sense of it. All right, so the question becomes, well, how do you actually get to a point where you are trapping single cells with a reaction buffer that you can then generate transcripts against many genes? And that's the technology, right? So again, the genomics core would be doing, the SMART core would be doing this end of it. And the answer to that is you actually works through a kind of a fluidic machine. And this is how it works. So here you have barcoded gel beads. All right, these are actually called your GEMs. All right, so GEMs stands for gel beads in emulsion. And this is where all of the reaction materials are located, okay? So then what you do is you send it through one opening on the machine. And then at the other end of this, you would have your cells or nuclei, plus some enzymes associated with that as well. And you would feed that through the other end of the instrument, all right? And so what would happen is that your cells, your single cells, would then come in contact with a single gem if everything is working appropriately. And what would occur is that reaction mixture would then be associated with your single cell or single nuclei. And it's in an oil type of environment. And as a consequence, it would encapsulate that, okay? So Brittany is going to make libraries of each single cell in the context of this machine, all right? You're going to collect these, and then she'd make libraries, and then each of these would be barcoded. And I'm going to show you kind of how unique the barcodes are on the next slide. Because what you need to do is you have to keep this sorted so that the transcripts of a given cell, you need to then kind of work your way back and identify that those transcripts came from that specific cell. And that's kind of where this barcoding comes into play. All right, so how does this actually look? So again, this is your reaction mixture, okay? And each of these have a barcoded oligonucleotide, which is gonna allow you to make cDNA synthesis along with a specific barcode against a given transcript from a basically labeled cell or targeted cell. All right, so how does this look? And again, this is all kind of kit-based, which is, good for that. But this is what you're asking the technique to do. So let's say you have 3 different cells over here. You have to have a barcode for that cell because you have to go back and figure out what transcripts are differentially expressed in that cell, okay? The mRNA is actually a cDNA here. It's corresponding to the RNA that came from that cell that is now barcoded. You will have a unique molecular identifier, which is marking that mRNA in the process of making cDNA from it, all right? So now what you'll have, so let's say that you have thousands of cells. Each of those cells is going to generate a barcoded transcript or corresponding to a barcoded transcript that has the RNA that corresponds to the RNA of interest along with a unique molecular identifier. All right, so again, it's a very kind of elegant technique, and it's something, in terms of building libraries and sequencing that we can do in the genomics core. All right, so what does this actually look like? And again, this isn't something to go home and do any homework about, but just kind of out of interest in terms of, how can you actually go read back of an RNA coming from a specific cell type? And the answer is barcoding. All right. So you have what's known as a simple sample index. Now, the sample index allows you to identify a sample. So let's say you have 4 samples. Well, when you sequence this, all 4 samples are going to be sequenced together. You need a way of pulling them apart. And again, the bioinformaticist will do this based on a sample index that labels those as being unique from a given group. Okay, so that has to be there. But the real magic starts here. So you have a 10x barcode, which is 16 bases, which allows you to then map that change in or that transcript back to a given cell, because each cell will have its own 10x barcode. So again, that to me is really where the magic happens. And then you have a unique molecular identifier, which would then go back to that particular RNA that you have captured and made cDNA from it. Okay, so that's the technology behind this. And again, this is what the core does by building your sequencing compatible, single cell locating libraries. All right, so I don't know how far I'm going to get, but I can at least show you one success story. And again, this is older because as these techniques have been around for a little bit longer, what's happened is these papers are getting exceedingly complex, and we just don't have enough time to kind of work through that. So I'm going to work through this one, and this is the actual title. If you're interested, and you can go back and look at it in further detail. But the reason I kind of selected is it does some kind of cool things that you could think about in terms of your single cell experiment. Now, their idea is they were interested in heart tissue, all right? And they had females and they had males. And what they were actually interested in was not the myocytes or the endothelial cells, because those types of cells make up a lot of what the heart is. They were in fact interested in cells that were not those cells. So they were interested in identifying some unique cell types that was in the heart. And that kind of goes back to what we were talking about initially. Why would you use a single cell approach? Potentially to identify some unique cell types that are relatively few in number compared to other cell types that kind of dominate that tissue. And so the reason I thought this was interesting is that they actually did some sorting before they actually made libraries. So, and that's what this is all about. So they're getting rid of myocytes, they're enriching for nucleated cells, and they're also reducing the endothelial population. So they're really kind of helping their experiment before they got into the very detailed analyses. And and this could be something that might be useful in your system as well. You would just have to talk to myself and Dr. Jim Denver, and we would kind of help you work through this. Okay, so what did they get? Well, this is kind of what you get. So, and this is bioinformatics, in terms of getting to this particular point. These clusters are based on the mRNA changes that would come from library prep and then sequencing them on a next generation sequencer, okay? So those transcripts would then be given to the bioinformaticist, and they would do some, fancy bioinformatics that I'm not going to talk about here, partly because I don't know all the details. But what I want you to think about is what they're getting out of this information, all right? So what's happening here is they're getting clustering based on the X and Y axis, all right? And from that, they can actually identify unique cell populations, in their case, in the context of cardiac tissue. So for instance, they're finding dendritic cells, which is, as you all know, an inflammatory type of cell involved in that pathway, granulocytes, et cetera. And the reason that these are showing up as different colors is that within a cluster, they express similar sets of genes at similar relative levels. So these are being compared really to the other cell expression profiles within the context of that tissue. They did some other kind of positive control kind of things that you might also be doing. So for instance, they knew that within a cluster of B cells, within that analysis I just showed you, that this particular protein or gene is increased, and you would do these types of things as well, kind of positive controls. Okay, with all that being said, and again, we'll see how far I get, but these are some of the issues or the reasons that a single cell gene expression experiment, fails up front. And it's really the tissue dissociation, the cell prep issues. These are the things that we would have to help you with as you work through your experiment. So dead cells, you can't have dead cells in the prep. And that's actually a tall order because when I prepare tumors into single cells, I mostly get dead cells. So you have to kind of think about ways to get rid of that. The reason is, you get ambient RNA contamination. So this would kind of float into those GIMs. That would disrupt, a lot of your downstream analyses. You got to make sure you don't have cellular debris because again, you have free floating RNA that would then get into the droplet. You would make cDNA libraries from that, and then that would be incorporated into your analysis, and you could see why that would be a big problem. You can't have doublets of cells. They have to be single cells or single nuclei. Because if you have a doublet that gets into one of those GIMs, what you're going to actually make is a mixed transcriptome. And you're going to have an artificial hybrid cell, and that, that's going to cause problems as well. The process of enzymatically dissociating the tissue is also a big problem because that induces artifacts as well. You can have cell type bias in which you have, very fragile cells that don't survive the dissociation. You're going to lose those. You could have very large cells that have a hard time getting into the droplet. You may lose those. And then you have cells that really adhere to the rest of the structure of the tissue, and you would lose those as well. And one of the ways that we know that, there's an issue on the bioinformatics side, but you don't want to wait to the bioinformatics tell you you have an issue. We need to, figure these things out before you make libraries and, do sequencing because it's very expensive in terms of per sample charge. The good news is, these are things that the core can help you with. We are not going to dissociate your tissue. We're not going to collect single cells for you, but we can help you in terms of the troubleshooting. Another thing that's kind of a limitation as you think about incorporating this into your experiment is this is droplet-based single cell RNA sequencing, which is what we do. As a result of that experiment that we talked about, the cardiac experiment kind of highlights one of the issues. Is, of the cells that they analyzed, they actually identified about 2,000 genes per cell being changed, right? So that's, actually a pretty strong underestimation of mRNA changes that could be happening in a cell or just, mRNAs in general. So it's thought that a typical cell expresses about 10,000 genes. So really, you're not getting a full transcriptome, all right? That's the idea. And, we know that, and we know that that's a limitation. And this is something they're trying to address, but it's just increment changes. So when you go in, don't expect to find a low expressed gene because you're just not going to identify it. You have to come in with the right question. If you're interested in cell identity and mapping, well, then it seems to work just fine. You just need 2,000 genes per cell to identify those plots that we just saw. You can get up to 3,000 to 6,000 with some deeper sequencing, but that would add, significantly to your cost. So just think about the question that you're asking, and then, we can help you identify what technique might work best for you. All right, so I do have about 10 minutes, which is excellent because now I'm going to talk about the last one, which is spatial transcriptomics. And again, this, the most important step in this whole thing is, what question are you asking? Are you asking a question that would work for spatial transcriptomics? And that's kind of here. So we started with bulk RNA sequencing. Here you're looking for, I think of more of a large scale type of experiment where you're looking for changes in gene expression in response to some agent. I think it works well for that. If you're looking for identifying unique cell types in a tissue, single cell RNA sequencing may work good for you. Now, if you're interested in looking at how cells interact on a gene expression basis, cell location, cell-cell interactions, well, the only way that you can identify that is if you keep the tissue intact, right? Because if you're interested in whether or not, this cell type is interacting with this cell type, and are they spatially in the same place, and are those genes changes occurring within this context, well, then you need to preserve that. And then spatial transcriptomics might be right for you. Workflow is extensive, okay? Dr. Chris Risher, not sure if he's on the call or not, but he has been doing a lot of this work with Brittany, and I think Faith is the other person involved with this. And they're becoming experts at it, so that's a good thing. So we have some kind of resident experts in this process. But this starts way back over here. So here you have to get the slide, you have to section your tissue in a way that you know how to do. You have to get this tissue onto a slide, and there's an instrument that will help you do that. And what occurs is that the mRNAs that are in that tissue, and again, this has to be kind of a fixed location. You don't want tissue obviously moving around, because this is where the magic happens in terms of being able to identify that mRNA came from that cell, is because it's lined up over the top of these oligos that will capture that RNA and hold it in, literally hold it in place and give it a barcode that allows you to then go back and identify that cell is where that mRNA came from. All right. So again, as a lab, we would, be involved with this. We'd be, rooting you on, but you would be sectioning the tissue. You would probably, with some, help from somebody, maybe Chris Risher's group or us a little bit, would help you kind of align the tissue with the probes. And then, once you get to a point where it's time to make a library, Brittany, who's in the core, would do that for you. She'll make libraries from this. There's a lot of QC involved, and you would do sequencing, and then, you would get some heavy bioinformatics support to basically put everything back together. All right, so I don't want to spend too much time on this, but again, really kind of how this works is that, again, it's all barcoding. You have a unique barcode that is coming off of this capture area. It corresponds to that particular part, that spatial location within that tissue of interest. Again, you have a unique molecular identifier that tells that is a unique mRNA transcript, okay? And that's important for preventing it's a way to subtract out doublets or duplicate forms of a given transcript that can happen during the library prep. And then you have the poly A that kind of capture that, okay? So again, your tissue is going to go on here. These spots or these probes are here to catch your RNA. And then when Brittany makes the library, she's going to attach these barcodes to it. All right. I don't have time to go through the details here, but just know that this is what's associated with that, magic slide. And it allows you to make a cDNA corresponding to the mRNA that came from the tissue that has a barcode that the bioinformaticist can use to relocate where that RNA is coming from. All right, so, what do you need to kind of do this? You need dedicated people, because again, this is not, these aren't trivial types of techniques, but you need a site assist, which we actually have two of them. I didn't realize that, but we have one. We have another one floating around. This is what allows you to line up your tissue appropriately with the slide that has the probes that allow you to then map your way back to where that mRNA came from. You need some type of next generation sequencer. So this is the NextSeq 2000 that we can use in our core to sequence the libraries that come from your site assist. Some things to think about, and as you set these experiments up, I don't know how much this is talked about any longer, but it's worth kind of mentioning these things up front. So when you think about classic Visium, and again, I'm not sure if that's what we do anymore, or if we just do Visium HD. But the distinction between these two is the following. It's just the size of the spot. The spot, again, is what your tissue is lying on top of. And you're using that to identify where the transcript came from within that tissue. Now, the problem with having a spot that's about 55 microns in size is you're not just going to see an RNA from one cell, right? The resolution's not that good, because you can have actually 10, potentially up to 10 cells that are contributing to that spot, all right? So you're actually getting an average signal per that region. And it's actually, my understanding is useful for tissue level features, okay? So now there's, that was recognized as a limitation. And then now they have something known as Visium HD, which is high definition, I suspect. And what they've done is they've reduced the spot size. And so now with our advertising, and again, I'll have to talk with Chris Risher and Luis Risher, how well this is actually working, that you can get down to a near single cell resolution now. Now, the caveat to that is that you need a lot more sequencing now to identify the transcripts that are, coming down and being associated with that tissue. And so it's a lot more expensive. I'm not going to go through this because I only got like a minute or two. I just want to kind of emphasize that complicated workflow is just to get to this point in time. So here, your bioinformaticist is going to run it through a number of different pipelines to take your transcripts, basically, your barcoded transcripts, and then map them back to where those transcripts are happening in the context of your tissue. I got a couple minutes, and again, these studies are getting more and more complicated, so I just kind of stole these from 10x just to kind of highlight, the kind of data that you're going to get. Again, just real briefly, it's going to cluster based on similar expression changes and similar level of expression into these clusters. And then within these clusters is going to show you, the types of mRNAs that are uniquely expressed at a given level within that cluster compared to your other clusters. And here's kind of an example of, the type of data or thought processes or the information that you can get from this. And again, I only have a few minutes to talk about this. If you're really interested in doing this, you would set up a meeting with, myself, Jim Denvier, and, Chris and Luis Risher, if they're able to kind of join in on this, to really kind of talk about, what they're doing with this type of analyses. But here, what they've done is they've stained for a marker of a differentiated neuron population by immunofluorescence. And what they're doing is they're tracking that through or overlaying that with their Visium whole transcriptome. And when you do that, you can actually identify or get down to, additional differentially expressed genes that are within that cluster. And, again, I haven't done these personally, but you can see kind of the resolution issue. You're not getting down to single cell per se, but, potentially with Visium HD, you could get a little bit better resolution. But it's a discussion that we would have before you really got into this is, what type of resolution are you comfortable with? Is this new HD technology, is it getting better and allow you to really pinpoint, on a cell-to-cell basis? All right, well, that worked out perfectly. So if you have questions, and again, that's really something I want to kind of highlight about this, is you really need to set up meetings with, myself, Dr. Denvier, and again, we may have to pull in other folks that, have some experience with either single cell. WVU has a core that is just really devoted to single cell spatial transcriptomics. They're excellent to work with. They have lots of experience. So, we would get and kind of work with them. Jim Denvier and his group would be, definitely involved in the conversations right up front to talk about the bioinformatics associated with doing this, whether or not the experiment is feasible from a bioinformatics standpoint and things like that. And we would be happy to meet with any of you to do that.